Yeah, we can.
Okay, thank you, sir. So, good evening, everyone. I warmly welcome all the participants and sincerely thank each one of you for joining today's roundtable discussion on the topic microbes mediated bioremediation. So, just let me introduce this topic within 1 or 2 minutes. So, we all know that the bioremediation simply it means using microorganisms. So, microbes means bacteria, fungi, and algae that we use to break down the harmful pollutants into the harmless products. And these microbes, like the act, we can say that they act like the natural cleaners. So they feed on the contaminants such as the oil, pesticides, plastics, dyes, sewage waste, and the industrial chemicals. And they convert the microbes convert them into the safer substances like the water and the carbon dioxide. The byproducts are there. So this method is the safe, cost-effective, and sustainable because in the last roundtable discussion, Halami sir has rightly said that we have to or conduct one session on the sustainable microbiology. So once again, I welcome you all in this roundtable discussion, and I look forward to the meaningful exchange of ideas, exchange of knowledge. So now I would like to request all the participants to share their research experience or any knowledge on the bioremediation. You are free to share here. So just unmute yourself and you can share your research experience on this particular topic, microbes mediated bioremediation. Hello, Dr. Rahul Khatri, Dr. Barsa Chauhan, Dr. Salija Diman. Okay. If you have any experience on this topic, research experience or the knowledge, you can share with the participants.
You can ask opinion of one by one only.
Sure, sir.
What topic they are working on, or whether they are associated on similar line in their research activities, what work they did or doing, any difficulty they find, so that it
So,
will be more informative for them and educative also. Because if guide is not very serious or guide if it is there in air
mhm.
conditioned room, then
Even
can
students, if you have any query regarding this
yes,
topic,
yes.
you may put here.
Because last 2 3 seminars that discussion was quite useful,
Yes, sir.
and we could give a quite good suggestion to them.
So particularly on that isolation of microbes last session we have discussed. So Akash Kumar, Mohan Kumar, just unmute
Good
yourself
evening,
and
sir.
introduce yourself here.
Good evening, sir. Myself
Good
Akash
evening.
Kumar, and I'm basically from Motihari. And
Okay.
currently, I'm pursuing a PhD from botany under
Mm-hmm.
supervision of Dr. Suresh Naran Mehta from SNS College, Bihar University, Motihari. So
Mm-hmm.
I'm currently pursuing research on phytochemical screening of bioactive extracts of Cucumis sativus against pus-forming bacteria. Means
Mm-hmm.
a lot of means in our body that is pathogenic infection like डिप्लोकोकस स्ट्रेप्टोकोकस जो हमारे बॉडी में जो भी इन्फेक्शन जो क्रिएट होते हैं तो
Mm-hmm.
कैसे जो कुकुमी सटाइवस का जो बायो एक्टिव एक्सट्रैक्ट्स यानी फिनॉल कीटोन्स टेनिन्स ये कैसे उसको पस फॉर्मिंग बैक्टीरिया में हेल्प करता है इनहिबिट करने में सेल वॉल को लाइसिस करके बैक्टीरियल सेल वॉल को क्या करता है लाइसिस करके
Mm-hmm.
दे कैन जस्ट ब्रेक डाउन द सेल वॉल एंड जो भी हमारा जो खीरा में जिसको हम लोग कुकुमी सेटाइवस बोल रहे हैं जो कुकुम्बर में पाया जाता है वो क्या करता है लाइसिस
Mm-hmm.
ब्रेक डाउन करके और उसको इनहिबिट कर देता है और पस फॉर्मिंग बैक के अगेंस्ट में बहुत ये अच्छा काम करता है
अच्छा
इसी
अच्छा
पे सर मेरा रिसर्च है बट आई नीड टू जस्ट डू द phytochemical for some of the bioactive extracts from cucumbers. I suggestion from your side, sir, regarding phytochemical screening, regarding
Mm-hmm.
regarding where I have to do phytochemical screening. Like
Okay.
last year, I qualified my PAT exam, a state university exam, and last year I enrolled for the PhD. Currently, I'm just pursuing
So
PhD.
Akash, we will take your means question at the last because today topic is little bit different.
Okay.
Bioremediation. Okay. So obviously we'll discuss your topic means question. Okay.
Okay,
before
sir.
end of this
Thank you
session.
so much. I
Thank
need
you.
to connect with on your WhatsApp also because I have to too much suggestion from your side.
Sure, sure.
Thank you so much, sir.
You can take my number, okay? My
Okay, sir.
WhatsApp number, you may contact me.
Okay, can I take from group? You are in group.
Just I am sharing it, okay?
Okay, sir. Thank you. Thank you, sir.
In chat box you can see it. Now the Dibya Rabi, Dr. Rahul Khatri. Just unmute and introduce yourself that so that you can know each other.
Yes,
Yeah,
I
yes,
am
yes.
Dr. Alphatri, and
Yes.
I have currently working as a guest faculty in Swami Shraddhanand College, Delhi
Mm-hmm.
University. And my PhD topic is not like related to microbiology or bioremediation. It is towards the physiological aspect at the high altitude, hypoxia on the human beings. Currently, but I am taking the subjects of the microbiology like environmental microbiology and microbes in environmental management. So I have joined this uh, discussion to know more about the role of the microbes.
Thank you, Dr. Rahul, for joining.
Just a minute, Dr. Khatri, in high altitude, what you are looking for, you said?
Sir, I am like because I am teaching the subjects in microbiology like environmental microbiology and microbes in environment management.
No,
So
no. In you told no, in high altitude you are looking for some specific physiology.
yeah, no, on that at my during my PhD, I have worked like the effect of the high altitude hypoxia on human
Ah,
individuals. So
yes,
we isolated one biomarker, which is the BNP, B type natriuretic peptide.
very
So
good.
I isolated that as a predictive biomarker to identify the hypoxia susceptible individuals at the sea level itself
Yes.
before exposure to the high altitude.
Just to supplement again, deviating from the bioremediation topic, even we have the DRDO project
Oh,
on
sir.
the soldier deployed at high altitude. That's all I
Yes,
can
sir.
tell you this moment. So remaining we can talk some other time. High altitude microbiology when Dr. Tiwari ji will organize, maybe that time we can talk the other topic. What changes happens in the gut microbiota from microbiological perspective.
Yes, sir.
That time we can talk. Okay.
Okay, sir.
Over to Dr. Tiwari.
Okay, thank you, sir. So now Alakpan Gandhi, Dibya Rabi, Dr. Barsa Chauhan. Just introduce yourself and if you have any means query or the info. Yes, Dr. Barsa.
Hello. Hello everyone. This is
Hello.
Dr. Varsha. I have completed my PhD on tuberculosis from Vallabhbhai Patel Institute. So
Hmm.
before this, I never worked on bioremediation. I am just here to know more about bioremediation.
Okay. Thank you, Doctor.
Yeah,
Thank.
just Dr. Tiwari,
Hello.
if,
Yes,
if
sir.
Dr. Varsha can tell what type of work she did on the tuberculosis, it will be interest of my lab because we work on a
Sure,
peptide
sure, sir.
which is actually activity against Mycobacterium tuberculosis,
Sir, I
antimicrobial.
worked on I worked on efflux pumps genes and
Okay.
see their roles in registration and
Yeah.
in resistance related to
Exactly.
delaminid.
Very, very useful topic. And then we know that Mycobacterium tuberculosis is a very champion in developing a drug resistant, excessive drug resistant, multi-drug resistance. This
Yes,
is
sir.
very, very important topic actually. Right.
Thank you, sir.
Exactly.
पता है कि डॉक्सन और आदिल रशीद बीच में डालेंगे तो फिर फकर जबान भी थे लेफ्ट हैंड बैटर
Hello. Dr. Rakesh. Hello.
लगा ही नहीं कि पढ़ पाए और ऐसे भी उस 2022 के फाइनल में आउट किया
Arula Jagan. Chitran
Yes,
Ramesh.
doctor. How are you?
Yes, sir. Fine, fine, sir.
Yes, doctor. I work on extremophils, as you know, from our previous meetings. So
Mm-hmm.
I work on these halophils, thermophils, alkalophils, and acidophils for bioremediation process focusing on industrial wastewater such as petroleum and phenolic industrial wastewater, as well as we work with also sludge reduction and then microbial fuel cells. This is the area of research we are focusing on. Currently, we are working on biohydrogen production. Also, with your support, after attending the AI sessions, I'm focusing on implementing this AI in reactor designs and how to optimize the conditions for operation of reactors.
So this AI is the new topic, sir. Can you elaborate a little bit, sir?
Which one, Doctor?
Use of AI in bioreactor, sir.
Yeah, this is like we have some microbial fuel cells, reactors. So we want to optimize that, how far it is matching with the AI as well as with the practical results and how it is AI oriented, how we can optimize the organic loading, how much amount of organic loading, because practically speaking, it is little bit different, but when you go with the AI, a slight difference is there, but it is almost matching with that. So we
Very
are
good.
trying to configure that.
Yes. No, but what is the organic matter you are using for the microbial cell?
Sorry, it's an industrial wastewater. Actually, we
That
use
is what actually my question is. Very much fit
yeah,
with this
it's
microbial.
like we use cosmetic industrial as well
Yes.
as organic rich industrial wastewater, something which is rich in high organic content, but currently we are
Exactly.
focusing on acidophils.
Exactly. Yes. Very much
Relevant
pertaining
to
to
this.
the roundtable discussion.
Yes, sir.
Because such kind of waste no one is treating or using for energy generation, which you are doing,
Yes,
right?
we are working on energy generation as well
Yeah,
as
that is
yes, treatment with for the industrial wastewater.
exactly where wealth from the waste, which you are converting.
We directly get the waste from industries. So we
Exactly.
visit the industries, we try to solve their issues because their wastewater is around pH 4 or 3.5 to 4. So we try to
Exactly.
get the wastewater and we try to give our remediation, and we also develop some sort of extremophiles and we try to employ them in the secondary wastewater treatment.
Okay.
Very useful topic. Very useful. If if
Doctor.
you can share some more information, is highly desirable.
Yes, sir.
Means how you are
Just
adjusting the pH, how what is the BOD level, and then how you are initially giving what treatment you give.
So also the
okay.
secondary treatment.
Exactly,
Uh,
primary treatment, secondary treatment.
That will
yes,
also
this
help
is.
our students if you explain.
Yeah, they are doing something like they collect the industrial wastewater and they take it to the treatment plant. Over here, it's happening, but usually now the industries they have to implement their own treatment plant over here itself, where they are placing the industries. But now what they are doing, they are just collecting this low pH because they cannot let it along with the other industrial wastewater. So they have to collect this low pH wastewater and take it to some other place where they can treat almost all the low pH industrial wastewater. So initially they do for the screening and try to optimize that for adjusting the pH, and they go for other treatments. But employing this kind of acidophils, they are very much interested now. So how they can get rid of this low pH, treating this low pH wastewater, and for also this we are at the lab scale we are focusing on energy production, but they want something else to do with those, whether they can recycle that, and there are many focus because we have to work with engineering oriented also, because the design and other things also stuffed in along with the when we work with the industries.
So thank you, sir. But sir, can you little bit explain that means for the student the secondary treatment so that they can start means
Secondary treatment is the
remediation project
biological
at the
treatment. Yeah, sure.
small level.
Secondary, yeah, sure. Secondary treatment is the biological treatment. Where
Yes.
they use the activated sludge process. This is employed with mostly with the microorganisms that will be alive. Aeration is provided with big aerators, and then this we have to measure the dissolved oxygen also there. This oxygen provides the growth and the nutrients from the waste. It is utilized by the bacteria present there, and they try to treat them, and from there it is further the process. It is like a schematic representation. You can see in any of the effluent treatment plants, you can say there is a collection tank, and then screening means they will remove all the solid waste, and which can be where there is a grid is placed, and it will remove the for example, if it is a coke tin or something like tin materials or whichever is solid material, they will be screened out there, and further this water is screened, after that pumped into the aeration tank. Aeration tank, it is already provided with, but prior to that, if you want, we can go for pH adjustments, and then we can go for the treatment process of activated sludge process, and further it is go for the settling tank. This is a basic one I'm talking about, but it depends upon each industries. They have different criterias of handling the wastewaters. So from this activated sludge, they go for the settling tank. From settling tank, they go for the they will try to remove the sludge materials, and further the treated water, they try to reuse it with a little bit of chlorination, and then it can be used for any irrigation or just a lawn or some other purposes. They are trying to use it over here. This is regarding the domestic wastewater, but industrial wastewater, we have many obstacles like pH, salinity, and also temperature. Over here, in summer, it goes up to 50° plus. So we have to have something like thermophils as well as halophils, halothermophils to survive and degrade the organic materials whichever is coming out of the along with the wastewater.
Yes, sir.
Very useful information. In between, my net was not correct, so I missed that, but not a issue. You have covered very important topic and what microbiology can really do to achieve the sustainable development goal, or whether sustainable development goal or not, but at least how we can keep our environment clean, and especially the sewage water, especially with reference to the irrigation water. Right now, most of the actually pollutant water people are using for the irrigation purpose, and those chemicals, pesticides, viruses, they are actually coming in our crop, you know. And these antibiotics, these are actually the traces has been reported. So if such effective bioremediation strategy, if it is exist or available, it would be really a big boon to clean the environment, to identify the possible pollutant, to understand the nature of this discharge. And this is what exactly just happened with reference to antimicrobial resistance, because several years we didn't bother. So we were just releasing the AMR bacteria in the environment. Now they started coming in food chain. They are in the environment. They are actually in the sewage water. They are in the your irrigation water. They are now in the plants, crop plants, as endophytes. So now AMR is not only in the clinical setting or in the veterinary or animal husbandry, but it is in the food chain also,
Yes, sir.
environment also. That become actually a big challenge for everyone. So finally, it is the microbiologist who has to answer. Finally, it is the Dr. Bipran's Tiwari who has to arrange the round table and find the possible solution, in other words.
This one is the objective of roundtable discussion, sir. So now, so one question, sir, Arula Jagan, sir. So your lab has developed any consortium, sir?
Yes, please go ahead. Yeah, we have developed already alkalophils. Okay, and then acidophils we have one, I think we have already published. It is on Stenotrophomonas maltophila. And in 2017 we published, and still we are working on some other consortia on acidophilic treatment for the petroleum also. And because this industrial wastewater, we have noticed, especially in the cosmetic industries. Recently, we worked on this cosmetic industrial wastewater, which is having low pH, and then the phenolic compounds are heavily polluted in that. So we have to remove that. And the COD level of this kind of industrial wastewaters are 30,000 plus.
Mm-hmm.
So we have to reduce those things, and then that's the biggest challenge they are facing. And you know very well, every year the standard has been increasing. We have to find a appropriate technology for treating those things. So,
Mm-hmm.
and it should be also a, eco-friendly. So
Yes, sir.
we have to go for an integrated approach. Something like a thermochemical and biological treatment, something like that. We have to focus on that. So we have to trial with different chemical, biological, or like physical, biological, something like that. We have to check with different combinations. So it will bring up something with novel technology for treating this kind of wastewaters.
Yes, sir. Thank you so much, sir. So now Chaitanya Ramesh. Are you there, Chaitanya Ramesh? Dr. Prasoon sir has joined. Dr. Prasoon sir. Dr. Saleja Dhiman. Kevin Joseph. At least because you have joined this session for 1 hour, so at least you can do that. You can introduce yourself. Snehal Jadhav Tejal.
Dr. Shailja wants to talk, I think, something.
Okay. Dr. Saleja Dhiman. Dr. Prasoon sir.
Yes, sir.
Yes, sir. You are audible, sir.
Yes, yes. Uh, I hope I am audible too.
Yes, sir. You are fine audible.
Yes, yes. It is so nice discussion actually with Arul sir,
Mm-hmm.
Arul Jagan sir. No, but I am still a bit confused with the previous methodology that was used. No, this uh, optimization strategy using box bank and design. No, BBD. People previously used BBD to optimize the scenario. So how different the AI is? This question is for Arul sir actually, because he is already working on AI based optimization.
We just started doing that, doctor. Actually, we just started to do that optimization using the AI, because I'm just a beginner in that, but I have some other persons who is really working on in the medicinal field. Okay, so
Okay.
I'm trying to just get the input from them, and then how far we can go with that. So we are working on that. So uh, if the output is good, then it will be great for us to optimize many parameters, because we design our reactor and we work on different industrial wastewater. So we need something before designing, as well as uh, there are many inputs. Yeah, I think you would have gained knowledge on. I'm not sure. ANN, I think. And then there are some other networks are there in AI. They are employed, but currently I don't have any slides to share with you, unfortunately. So, uh, so whatever coming into my mind, I'm just responding to you.
No, no problem, sir. That is fine because we are here to discuss now. Actually, this question is always coming in my mind,
No,
especially
this is
regarding.
a model, Benbox model, and other things. You get this data and you try to do implement and incorporate this data, and then you derive the models and how it is fitting into those models. But this is design oriented. How far, if you are having something like how much is the organic loading rate and hydraulic retention time, and what is the feed amount, something like that. Protein concentration of the feed amount means the biomass concentration. Everything we have to give as an input, and it automatically we try to employ something on the Python. So, and we try to derive based upon that how far it is working. So I'm just working with the other person who can make this happen.
Mm-hmm.
So if I get that done, maybe I'm not sure how long it will take, but in future I can make it as a presentation for you.
Sure, sir. We would be looking forward to that because one time maybe Vipran sir, one time we can
Yes,
put
sir.
this
May
like
organize one dedicated
yes,
session for
yes, like
presentation.
AI in microbiology or something like that. No, or maybe just AI in bioremediation.
Sure. All credits goes to Dr. Vibresh Tiwari because he is the one triggered us.
Yes. He has always conducted such meetings, and we always get new idea, new thought process by discussing such things.
Yes, sir. So now, thank you, sir. Now,
Doctor,
Dr.
yes.
Yes, sir.
Dr. Sanjay has joined. Please go ahead.
Dr. Sanjay and Suryavanshi. Dr. Sarija Dhiman. Dr. Sarija Dhiman, are you there? You're not audible now. Dr. Sarija.
I asked her already. I think she is not available.
Hello, good evening, sir.
Dr. Good evening, sir. Madam.
Good evening, sir. Sir, I'm audible now?
Yeah, you're audible.
Yes, sir. So, my name is Dr. Shalja Dhiman, and I am assistant professor in Sigma University, Vadodara, Gujarat. Sir, I have worked on biocontrol of fungal diseases through metal nanoparticles, which we have synthesized from green process. So this was my PhD work, and then I have worked on hydroponics also. In hydroponics, I have used chitosan nanoparticles. So currently, I'm working on hydroponics. I have started. I'm also working on Hello files the sir was talking about. I am looking into the picture of its antimicrobial potential against multi-drug resistant pathogen. I'm just going to start what I will choose, the plant pathogenic microorganism or the human pathogenic microorganism, because there is a very uh, tedious process for ethical clearance also. So, sir, I am here because of one of my colleague. She is working on microplastics, degradation microorganisms. She is a little bit confused how she should proceed next, actually. I think she is available here, Miss Nadiksha.
Nidicha Patel.
Yes.
Yes, he is present.
She is PhD student and she is working on microplastic degradation. She was asking me the questions. I suggest her to ask here her queries.
Nidicha, please unmute yourself.
Hello,
Hello.
hello everyone. Myself Nidiksha Patel, PhD scholar from Sigma University, Vadodara, Gujarat. My PhD topic is biodegradation of microplastic using microbial consortia which enhanced by green synthesized nanoparticles. Sir, I am on a isolation part of microorganisms. I have isolated 8 microorganisms, which includes fungi, and 7 bacteria. So I am a little bit confused against how I can make a consortia of that microorganisms, like how much combinations I will make, so which will give me a better result to degrade the microplastics.
Also the methods how she can look into the degradation part, how she can
Yes.
observe that the degradation happens or not.
Dr. Alami, sir. Hello. Prason, sir.
Yes, sir.
Do you have any experience on these topics, Prasanth sir?
Yes,
Plastic degradation.
making mix with
I'm
uh,
Yes,
trying
sir.
to connect to my tab, but interesting topic, very interesting
Yes, sir.
topic. Can you hear?
Yes, sir. Now it is audible, sir.
Can you hear me?
Yes, sir. You are audible, sir. Now.
Yes, sir.
Yeah. Prasun can tell.
Prasanth sir.
G. I have means worked on microbial mixed microbial consortia.
Mm-hmm.
Basically defined just like C has isolated, I think Nixida, right? So C has isolated some 8 microbes, 2 fungus and 8 microbes, right? So
7
on the
bacteria.
yes, bacteria. So the to start with, I think first is to check the compatibility among the different microbes that you can do using the t-test. So you just put some agar and use a streaking method to make a tea. And if they are not inhibiting each other, they are going together, then there is no problem. You can go ahead with making the consortia. However, while working with consortia, the growth stage is also a limitation because some organism will grow faster than the other. Okay, so whenever you are putting some microbes, their growth kinetics should be almost matching. Okay, it may not be exact, but it should be like relevant so that at least for 4 days or 5 days you are making any kind of treatment, then they all should be in good number. And although it is a labor intensive process and it will require all type of combinations to test. So you may begin with 2 microbes at a time, 3 microbes, then four, and keep checking on their abilities, at least from the growth profile perspective. Then you may go for degradation studies, microplastic degradation studies.
So her question is how she will means get information that plastic is being degraded in presence of the microbe.
That aspect, sir, even I am not sure because I prepared microbial consortia for the product formation like
Hmm.
hydrogen or PHA. Here the question is for degradation. So if so far I understand regarding plastic degradation, if it is microplastic, if it is soluble in something, you have some standard molecule, then before and after treatment you can measure the amount of bioplastic
Hmm.
in the solution.
This one is the primary one.
Yes, yes.
Measurements. And
Measurement,
is
yes.
there any production of there must be the production of CO2, sir, during the degradation process.
Yes, but then it will require a gas chromatograph, na?
Yeah. Hmm.
That also becomes difficult. Even if you are putting some kind of carbon source, so means the measurement, the standardization will take a bit time. While
Hmm.
the gravimetric analysis or some other method of microplastic detection can be used, means direct
Hmm.
method. Yes, that will
Okay.
help a lot compared to indirect methods like gas analysis. This is my opinion, although those who may have worked on microplastic, they may also shed some light. Regarding consortia development, there are again some statistical tools are also there. Like I used Placket Berman design to prepare the consortia after the
Okay.
compatibility test.
Can I add on microplastic degradation by consortia bacteria discussion?
Yes, sir, please.
Add on microplastic degradation by consortia.
Consortia bacteria.
So I think this is what we were discussing, no?
Ambarish, would you like to share means your experience? Any?
Uh,
Ambarish
yes.
Sindagi. Hello.
Okay, she want to add something to this.
Yeah,
Yes,
yeah. That's
Ambarit. Yes, please go ahead.
Ambarish Sindagi. Please unmute yourself. Hello. Okay, I think that network is not working here. Dr. Prakash Salami, sir. Hello.
Hello.
Hello. You are audible
Can,
now, Ambarish. Yeah.
am I audible?
Yes, yes, you are audible. Fine.
Sir, myself is Ambrish Chandni.
Mm-hmm.
Myself is working on bioremediation of some heavy metals, cadmium. Currently, I am about to submit my thesis. So I
Mm-hmm.
will add on bioplastic degradation, which discussion is going on. So
Mm-hmm.
what my suggestion is plan for minimal media,
Mm-hmm.
target bacteria which say isolated.
Mm-hmm.
Patel, the candidate yes, no. So individual bacteria first you have to go for the growth kinetics. Individual bacteria, so you can have the ideology of the growth kinetics of that individual bacteria first of all. Then you can plan for the
Mm-hmm.
consortia in growth condition, optimize the pH, temperature, and all the growth parameters which are there, and see the changes which how the bacteria is going to acclimatize at that environment and how far it is degraded the plastic. So this kinetics will be going to work out for her studies. So until and unless you plan, design, optimize your media, you can't judge how far that bioplastic is going to degrade, or you can change the when doing the optimization, you can change the certain inorganic salts or inorganic factors which are adding to the media. Especially these are not going to work on the general purpose media like nutrient agar and all, right? So
Mm-hmm.
soybean cage in diaster and all, they are there. But you can plan for the minimal salt media or some stored medias are there. They are specially designed for the biodegradation of plastic, heavy metals and all. So that minimal salt media and acetate minimal media, there many are there. Many this one, many medias are there. Even Bushnell hash broth is there. So that also supporting for the degradation aspects. But I got some conclusion from my research on the bioremediation of heavy metals.
Mm-hmm.
So that same will be working for the bioplastic degradation. So later on they can go for some JCMS and all for the analysis of bioplastic. That is secondary. Even you can go for some spectral identification. That is the other thing. But the optimization is a very prime requirement for her. This one, whatever the methods she used to select for this one. No. So this is my add-on to this discussion.
Okay, thank you so much.
Yes, sir. Welcome.
Sir, Shailesh Dabe sir has joined it. Mohan Kumar.
Uh, Hello, sir.
Yes, madam. Dibbera.
Thank you so much, sir, for the clearance. Sir, I have
Mm-hmm.
one more question. If
Mm-hmm.
I have just started working with the Hello files, and I'm just looking for a, multi-drug resistant pathogen, which
Mm-hmm.
for which I should target. There are many experienced researchers are present. I just want to ask any multi-drug resistant pathogen in plants on which I can work if they can suggest me.
Plant pathogen.
Yes, multi-drug resistant plant pathogen, which is a very like harmful for our crops, so that I can work for them. Is there any?
Okay. So now if anyone working on the plant, so that
I'm
means you
not
have.
sure.
Yes, sir. Please. Yes, Prasun sir.
Yes, just I'm not sure about the multi-drug resistance, but so Ralstonia species, few Ralstonia species are quite peculiar. Ralstonia solanacearum kind of family. They Solanacearum family they infect.
Mm-hmm.
Means potato plant, tomato, and even few brassica species. So those kind of pathogens can be targeted, but their AMR status we need to check. I means their multi-drug resistance profile we need to check. Mostly people use pesticides, just pesticide. No,
Mm-hmm.
there are some microbial things people are putting that I am not sure. But these are some of the studies. There are some of the studies on the biofilm formation and how fast they go from the root to the tip or leaves. Uh, so they migrate very fast. So they are quite unique pathogen, plant pathogen that she may start working on.
Okay, sir. Thank you, sir.
Hmm.
So you have developed some drug or hello?
No, sir. Sir,
Mm-hmm.
I'm looking for some antimicrobial compounds from the halophiles, which can
Okay.
be effective against these microorganisms which are multi-drug resistance.
So why you are interested in plant pathogen only?
So, because there are very ethical concern for human pathogens which are multi-drug resistant, so that's why I was thinking that I should work on plant pathogen till that time.
Okay, thank you. So I have shared one feedback form. You can fill out that. Mr. Tejas has also joined. Mr. Tejas, he has submitted his PG. Hello.
Yes, sir.
Yes, Mr. Tejas. Just introduce yourself first.
Yeah. Yes, Tejas Chaudhary. Uh, yeah. I have participated in the last meet. Myself,
Hmm.
Tejas Chaudhary. I am currently working on microbial enzymes for the valorization of lignocellulosic biomass into various bioproducts like bioethanol, biogas, and other products. So I'm working at the Department of Microbiology and Biotechnology at R.C. Patel Arts Commerce Science College, Shirpur. Uh, yeah, I'm interested in this topic. That's why I am joining. I'm joining from last two lectures.
Mm-hmm.
Yeah, that's it.
Okay. So do you plan to work on this topic, bioremediation, in future?
Uh, yeah, actually, somewhat my topic also includes the bioremediation. Actually, I'm working on lignocellulosic biomass. So that is basically considered as a organic waste that is dumped into the soil. So basically,
Mm-hmm.
I'm also working on bioremediation, but not to the wastewater treatment, but to the soil treatment. Yeah.
Mm-hmm. Okay. Thank you, Mr. Teja.
Yeah. Thank you.
Now, Sailash Dave, sir, Thanushika, Badhiya Maitri. Are you there, Badhiya Maitri? Okay.
Hello, sir.
Hello, hello.
Hello, sir. I wanted to introduce myself too. Myself,
Mm-hmm.
Divya Ravi, currently working as Junior Research Fellow under the guidance of Dr. Deepa Srivastava ma'am in Deen Dayal Upadhyay, Gorakhpur University.
Okay.
This is my second year of PhD, and my
Mm-hmm.
topic is histopathological and molecular characterization of viral infection in Momordica carinthia, Bittergourd,
Mm-hmm.
in Eastern UP. Sir, I have
Okay.
done survey in some Eastern UP area like Gorakhpur,
Mm-hmm.
Kushinagar, Varanasi, and also did TEM, transmission electron microscopy. But
Mm-hmm.
I found there is a large entity pictures, but I don't know whether it is a virus or protein aggregates. So
Mm-hmm.
I also have to do like PCR. Sir,
Mm-hmm.
so I want to know that which diagnostic method would be most reliable for detecting mixed viral infection in Momordica carinthia, like RT-PCR, qPCR, or NGS, or what? Which method is most suitable for to confirm that
Differentiate.
whether it is virus? Yes, sir.
Hello. So anyone is
Yes, sir.
present who is working on the plant virus? So Dibbar, I have shared my WhatsApp number. Okay, you can put your
Okay, sir.
question there so that I can share with the expert. Okay, I
Okay,
think
sir.
there is
Okay.
no any expert on that particular because today's topic is bioremediation. But I will
Okay, sir.
share with the expert. Okay, because
Okay.
we have
Okay,
the
sir.
expert in contact.
Okay, sir. Okay, sir.
Thank you, Dibbar. So I
I
have
would like
shared.
to just
Thank
suggest
you so much, sir.
here
Mhm.
to the
Yes, Prasun sir.
yes, yes, just a suggestion. I am also not the expert, like you said, but
Mm-hmm.
just a suggestion is that if you know what kind of viruses often infect the plant you have chosen, then there are specific genes for the respective viruses, especially capsid proteins or polyhedron kind of thing, or coat protein. Na? So primers is specific to those specific viruses can be used. If you are looking for one unique virus, you may get the primer for that, and then do even RT-PCR will also help. Just like previously RT-PCR we have been using for COVID, right? To get to know about their number or the
Yes,
particle
sir.
through CT
Yes, sir.
value.
Yes, sir.
Yeah, yes.
Yes.
But you need to know what actually you are looking for.
Mm-hmm.
If you don't know, then it becomes difficult. Then it is like a hit and trial. So 2 or 3 rounds we need to go, or maybe you can purify the virus. No, you can purify them through
Yes.
dilutions,
Yes, sir.
and then check how many different types of plaques that are being formed, and isolating them and going for TEM to characterize each and every type of viruses that is infecting your plant.
But now it says it's result only from the team, no?
Yes, he has gone for TEM, but
Yeah.
also TEM data may have revealed if the viral morphology is different, then based on
Yeah.
morphology, you can know how many different viruses are there. Mm.
Okay, so now Prakash Salami sir has joined. Hello, am I audible to you, sir? Prakash Salami sir.
Yes, yes, there was actually some network issue with me. I tried to connect with my tab, but
Yes, yes, sir.
it did
Now
not
I would
work.
like
Now
would
suddenly we
like
came
to
to the
hear
virus.
from.
I could hear that last few points about the virology.
Yes,
So
sir.
that was also a important aspect in this, to determine the sewage intensity, how much is the viral population load is. From that angle, this is also important aspect. Any area which is not covered or which I can put forth my idea, or
I think
time is almost up?
yes, sir. That Nidicha Patel.
Uh-huh.
Are you there, Nidicha Patel? Hello.
So I will tell meanwhile she can come back and respond
Yes,
with
sir.
reference to okay, she is there. Please.
You can put your question. Okay, Prakash Salami
Yes.
sir is here. Hello.
Hello, sir. My question is, I have isolated the 8 microbes, which includes 2 fungus and 7 bacteria. So I'm looking forward to make a consortia. So how can which combinations I will make so that microbes will give a better degradation?
Degradation of what?
Degradation of microplastics.
Wonderful. So you can use the plastic or monomeric unit of a monomers, plastic monomer also, and you see that it is utilizing, right? Because the microorganism, when it is growing, means it should utilize those plastic monomer, uh, and then you can actually consider that this consortia. First you can actually check the initial, this individual one, then you can go with the combinations, see that both the bacteria are growing as the Dr. Pratush told, cross streaking or T cross test, right?
Yes, sir.
Or you can just combine it. You can first check the individual colonies, individual culture either in a liquid broth or in a plate, mostly in the liquid broth. where glucose or sugar is depleted. That means you will know that this culture is utilizing or not. First is the monoculture, and then subsequently in mixed culture, and then you can go on mixing 2 cultures, 3 cultures like this way, and whether you can see whether they are utilizing those plastic or not. How? Based on the growth, right? So that will actually give you a rough or preliminary idea that the cultures which you have selected is growing. Now, what was the basis of selecting those culture? You should be aware. Randomly, if you say select, then you will have the random results. Then subsequently, when you are actually checking these microplastics, you will be analyzing how whether it is degrading or not, right? So first is the growth checking, ensuring the growth that in the presence of glucose or a complete media it will grow, but minimal media with a microplastic or any plastic monomer, it may grow or may not grow. So if it is a growing, that means you have some sort of fair indication that the cultures are utilizing those microplastics, right, by the degradation.
Yes, sir.
Yes, one or if you are looking only whether it is degrading or not, then another method is the looking for some enzyme activities. You know that PETase is the enzyme which is identified, I think, from this bacteria only, I guess, which is actually hydrolyzes the this polyethylene, right? So if you get such kind of culture, then you have really have a very good chance to utilize even the single culture for degradation purpose.
रोटी में
Right?
टाइम लगता है ना अभी तो बोल रही थी हट जा हट जा हट जा
So PTL degrading culture,
रोटी
which is
बनी
actually published, I think 10 years back in the science, which is actually a very novel study because that is only the culture or microorganism which produces a unique enzyme, and that enzyme is responsible for eating away the plastic, which is otherwise xenobiotic or not possible to hydrolyze with any other, not possible through the microorganisms, in other words, right? And
Yes,
then
sir.
you can actually, once you have such a monomeric unit, then you can actually check by the GC, or FTIR, how the degradation is happening, right? And then actually you can develop the further subsequent study. So you basically need some sort of the polymer chemistry expertise also to some extent, the polymer science, and then you should have a good analytical knowledge in addition to this microbiology. Definitely you will get the good results. I hope I could be able to answer your question to some
Yes,
extent.
yes. Yes, sir.
Right.
Thank you so much, sir.
Yeah.
Add one thing to this. Hello.
Hello. Yes,
Yeah.
sir.
Yes, sir.
To add one thing to this question, actually, I have worked on microbial consortia in my PG project. Before forming a combination, uh, I suggest you first go for a compatibility test because that was the problem that I have faced in my PG dissertation. Whether the organisms are compatible with each other or not. So you just have to grow them on a single plate, and you have to see whether they are growing in the presence of other organisms or not, or else they will inhibit each other's growth, and it will not be called as a consortia. It will be a single organism. So yeah, that was my point to this.
Yes,
Yes,
yes.
sir.
When
Thank
you
you.
are mixing the culture, we have to actually ensure whether only one culture is growing or both the cultures are growing, or they are, rather than doing the actual purpose what we want, they will be inhibiting each other.
Yeah.
So that
Yeah, you will
compatibility test is very much required. But first,
combinations.
you can check the individual culture.
Yeah.
Then the combination. Once you get the possible good result, look that they are compatible with each other. And once it is a compatible, then you can expect a very good results. That is what actually, or you can actually change the ratio also. Some S to 1:1, or 2:1, or 1:2. So that ratio also will actually able to give you a very good clues with reference to when the degradation happens, what results you are getting is are convincing or not.
Yes, sir.
Yes.
Thank you so much.
Okay. So now come to the end, reach to the end of this session, today's session. So thank you everyone for your valuable time and participation. My special thanks to all the contributors. Dr. Prakash Salami sir, Dr. Prasun sir,
Thank
Dr.
you.
Arula Jagan sir, Nidicha Patel.
Thank you.
Arula Jagans actually information sharing was very useful to the topic
Yes.
focus.
Sir,
Though
we
we
would
also
like to hear from means one dedicated session we can conduct for him.
exactly. So he should prepare. He should
Yes, sir.
tell about his research activities.
Sure,
Not,
sir.
not a theoretical knowledge. And then
We are working practically, but I have to find time to prepare specifically for
we
it.
know we know you are quite busy person, so that's why actually we are inviting you to give a talk directly. You know.
Thank you
And
very much, Doctor. Thank you.
your knowledge definitely will be inspiring and useful from our audience perspective also, because we need such a practical knowledge. You know. Which you will be
Doctor.
able to share. So you have to spare some time, in other words.
Sure, I will try.
Very
Try my
good.
best. Sure, Doctor. Thank you.
Once again, thank you everyone for joining today's session. Thank you so much.
Thank you, everyone.
Thank
Thank
you.
you, sir.